Hello everyone, welcome back to our channel. This is Science Lectures 256. Don't forget to like, subscribe, and follow us on our social media handles at X at Science Lectures 256 and on WhatsApp at Science Lectures 256. So today we are continuing with our discussions about algae and applied phycology. So we are still looking at algal taxonomy, and today we are going to look at a few algal classes. Um, the current algae classification was designed by a man called Round in 1973, but his current algal taxonomy was a modification of the classification by this scientist called F.E. Fritzich, which he designed in 1935 and modified it in 1945. And his basis of classification of algae was the food reserve material, flagellation, and pigments. So he ranked algae as a division and classified it into 11 classes. It was supposed to be the most authentic and comprehensive classification then. Then in 1973, Round came back and reorganized uh, a Fritz classification because he had divided algae into ranks, and he gave these ranks phyla names. But he also added the presence or absence of a true nucleus alongside other phylogenetic relationships. So whatever we're going to discuss, these are going to be our points of emphasis. The algae, the pigment, what food material are stored, and whether is it eukaryotic or prokaryotic. Maybe the other thing that has come up along the way is the mode of reproduction. So, um, so we are going to begin with the first division, which is rhodophyta. This group is made up of what we call red algae. So many members of this division are marine, and they have the ability to live at great ocean depths more than other algae. And their thallus ranges from being simple unicellular to complex multicellular, and they exhibit a high degree of epiphytism. That means they grow on top of other algae, while others are parasites. Examples of the parasitic forms include Polysyphonia lanosa, whose host is Ascophyllum nodosum, then Serratium condicola, whose host is Codium fragile. Lithomanium and Rhodocotton are some of the epiphytic genera of red algae. What are some of the characteristics? Number one, the members grow by either anapical cell or by diffuse growth. So we shall discuss more of this when you're looking at reproduction. Number two, they are non-flagellate. That means they don't have any flagella. When we're discussing criteria to classify algae, we said members of xanthophyta and rhodophyta are the non-flagellate phyla of the algal division. Then number three, their cell wall is differentiated into two parts. The outer part made up of pectin, and the pectic layer is the one from which they extract agar. We discussed the importance of agar. Then it's also made up of carrageenan, and agar and carrageenan make up 70% of the outer part, and polysulfate esters. And I remember I said agar is extracted from members of rhodophyta, specifically from the outer part of their cell wall. Then the inner part consists of cellulose. The inner part consists of cellulose. Another characteristic is they have septa, and the septa has pit connections, and these pit connections allow continuous movement of the cytoplasm. So as the cytoplasm keeps on streaming, that enables exchange of materials. Then the shape, size, and number of their nuclei are highly variable. Some of them have the nucleus that is located within the center, and that nucleus is many times spherical, while others have their nucleus located within the periphery, and many times that nucleus is elliptical. Then they have one stellate star-shaped chloroplast with a single pyrenoid or many discoid chloroplasts with no pyrenoids. What does that mean? I'm going to use this illustration to show you what a discoid chloroplast looks like and what does a stellate chloroplast look like. So this is an illustration of Spirogyra. This is an illustration of Clinodomonas. I need to first make a disclaimer. These are not members of the group Rhodophyta, but I put them here so that we can understand the nature of the chloroplast. So this is a discoid. This is a discoid chloroplast. No, I mean this is the discoid chloroplast. This one here. This is the discoid chloroplast, and you can see the pyrenoids. Even this one has the pyrenoids. So some of the members of Rhodophyta have such a chloroplast. The chloroplast is like in form of strands. It is discoid, while others have such a chloroplast that is stellate formed. So then their photosynthetic pigments are chlorophyll A, and we said members of Rhodophyta have chlorophyll D, while others have alpha carotens, they have beta carotens, xanthophytes, and biloproteins. Just a reminder, we said biloproteins are found in two algal classes, phyla, that is Rhodophyta and xanthophyta. So these ones have phycoerythroid, and as you hear erythroid, so there is an aspect of red. So the red color masks the green color of chlorophyll and making them appear red. Then their food storage products are fluoride starch and galactoside. Then they have a highly specialized mode of reproduction. They have non-motile male gametes called spermatia, and the spermatia are produced in what we call the spermatangia, while they also have an egg produced in what we call a capogonium. And the capogonium, the egg is found at the base of the capogonium, while the capogonium has a long structure called the trichogyne. I think I have a structure here that explains that. Yes. So we said the male reproductive gametes are called the spermatium. The spermatium are produced within the spermatangium. Yes, they are produced within the spermatangium here. Then the female reproductive structure is the egg, located within the capogonium, and the capogonium has an elongated neck called the trichogyne. So we go back. However, they also undergo asexual reproduction by producing what we call monospores. A monospore simply means a large spore, a single large spore produced in a monosporangium, and also produce what we call neutrospores. The neutrospores are simply spores produced by direct division of vegetative cells. Then some red algae have extracellular deposition of calcium carbonate in their cell walls, and science has shown us that these algae that have the deposition of calcium carbonate are highly effective at photosynthesis. And these algae, since they have calcium carbonate, they are also components of the skeleton of the so-called coral reefs. So the red algae phyla is divided into two classes, bangiophycidae and rhodophycidae. So bangiophycidae has only one order, which is bengiales, and bengiales has genera like Bangia and Popophyra. Then class rhodophycidae has 6 orders, nominaliales, which has Nomalion and Brattochospermum. It has Gelediales, which has Gelidium, and what we have here shows how Gelidium looks like. It has order Gigantinales, which has Gigantina, then Cryptonomaliales, which has Cryptonemia, then Rhodomeniales, which has Rhodomenia, and Ceremiales that has Cerememium. Okay, so we need to look at the reproduction. Remember, we said we shall also look at some of the modes of reproduction in a few given classes. So we shall begin by looking at reproduction in Rhodophyta. We said that the rhodophyta has main reproductive structures called the spermatium. The spermatium are produced within the spermatangium, and it has few reproductive structures called the carpogonium. The carpogonium has an elongated neck called the trichogyne, and at the base of the carpogonium we have the female nucleus. So what happens? After this spermatium are released from the spermatangia, they will be carried by water. They will swim up when they come into contact with the trichogyne here, with the trichogyne. So what happens? The point of contact between the spermatium and the trichogyne dissolves the wall here at their point of contact, and then the male nucleus will move down the trichogyne up when it fuses with the female nucleus. Yeah, so to be carried passively by water, then it will swim up when it reaches the trichogyne. Then where the two meet, the wall will dissolve so that the male nucleus of the spermatium can move up when it fuses with the female nucleus. After that, the trichogyne will shrivel or it will disappear. I think I need to emphasize is this, that the carpogonium is produced on the carpogonium branch, while the spermatangium is produced on the anterior branch, and many of these come from vegetative cells. Okay, so what you've just discussed is general for both classes, bangiophycidae and floridiophycidae. They both undergo that what we have discussed, but the post-fertilization changes differ from bangiophycidae and floridiophycidae. So that is what we're going to discuss. So after formation, we've said the gametophyte or the vegetative cell will form the anterior branch and the carpogonium branch. The anterior branch will produce the spermatangium. The spermatangium will form the spermatium. The carpogonium branch will form the carpogonium, then the carpogonium will form the ovum. The two fuse together by fertilization or syngamy, form a diploid zygote. So in bangiophycidae, this diploid zygote will undergo meiosis, and after meiosis it will produce four haploid spores, and each spore is called a carpospore. So after the zygote will undergo meiosis, produce four haploid carpospores. Then these carpospores on liberation, they will germinate to form a juvenile. They will germinate to form a juvenile thallus, a small juvenile thallus called the xanthophyta. Then it will be this xanthophyta to grow, form a new algal body. Then the algal body will undergo differentiation to form the gametophyte and the cycle continues. So because we have two distinct haploid phases, this is the first one, and this is the second one. We have two distinct haploid phases and one distinct diploid phase. So because there are two haploid phases, we call this a haplobiontic, but we have also one diploid phase. So in total, bangiophycidae has three phases. So it's called a haplobiontic triphasic life cycle because of the two haploid phases and one diploid phase. Next, I want us to look at floridiophycidae. So floridiophycidae also has a similar structure. You can see here we have the vegetative cell forming the anterior branch and the carpogonium branch. The anterior branch forms the spermatium and it also forms the spermatangium. Then the carpogonium branch has the carpogonium that forms the trichogyne and the female nucleus. So we said the spermatium, when liberated from the spermatangium, will swim towards the trichogyne. The two at the point of contact, the wall will dissolve. Then the male nucleus will move up when it fuses with the female nucleus. So what happens post-fertilization? So just repeat what I've said. The male gametophyte forms the spermatangium. Male gametophytes form the carpogonium. This one contains the ovum, this one contains the spermatium. So when the two fuse, we form a diploid zygote. Haploid plus haploid, you get a diploid zygote. So after that, after that, the zygote will undergo mitosis. It will undergo mitosis. Remember in bangiophycidae, we said the zygote undergoes meiosis, but here the zygote undergoes mitosis. And when it undergoes mitosis, it forms a specialized filament called the gonymoblast. It forms a specialized filament called the gonymoblast. So this gonymoblast um, has a terminal cell. Remember, we said growth is either diffuse or it is either by an apical cell. So the apical cell of the gonymoblast produces the carposporangium. Just to take you back a little here. Yes, now you can see here. After formation of the zygote will undergo mitosis to form the gonymoblast. The apical cell or the uppermost cell of the gonymoblast forms the carposporangium. So the gonymoblast and the carposporangium forms the carposporophyte phase of floridiophycidae. The thing I forgot to mention is that the carposporangium now contains diploid spores called carpospores here. So the carposporangium along the gonymoblast form the carposporophyte, and the carposporangium contains diploid spores called carpospores. Go back here. Yes, carpospores. And the gonymoblast and the carposporangium form the carposporophyte. We say the carposporophyte contains the carposporangium that forms the carpospores. So when the carpospores are when they are released or when they are dispersed, they will germinate to form an intermediate structure called the tetrasporophyte. Then the tetrasporophyte contains the tetrasporangium, and this tetrasporangium will undergo meiosis to form tetraspores. Then these tetraspores, two of the tetraspores, this one and this one, will form the male gametophyte. Then these other two will form the female gametophyte. So that is how floridiophycidae, those are the post-fertilization changes in floridiophycidae. The male gametophyte forms the spermatium, female forms the ovum, the two fuse together to form a diploid zygote. Diploid zygote undergoes mitosis to form a structure called the gonymoblast. Then the apical cell of the gonymoblast here forms the carposporangium. The carposporangium contains diploid spores called carpospores. So the carposporangium and the gonymoblast, both of them form the carposporophyte. And when these carpospores are liberated, they will germinate from what we call a tetrasporophyte. Then the tetrasporophyte forms the tetrasporangium, which undergoes meiosis to produce haploid tetraspores. And the two of the haploid tetraspores form the male gametophyte, then two form the female gametophyte. So because you have two distinct diploid phases, and we say this is a diplobiontic, and one haploid phase, triphasic life cycle. So members of rhodophycidae have a diplobiontic triphasic life cycle, or you say those ones, or rhodophycidae have a haplobiontic triphasic life cycle. For them, they had two haploid phases and one diploid phase, but members of rhodophycidae have two diploid phases and one haploid phase. Okay, so we need to look at then the differences between members of bangiophycidae and floridiophycidae. Members of bangiophycidae have less complex sexual reproduction, while those ones of rhodophycidae have very complex sexual reproduction. Then number two, the pit connections, we said they are found in floridiophycidae, not bangiophycidae. Then the calcium carbonate is deposited in members of floridiophycidae, not bangiophycidae. Then bangiophycidae is found in freshwater, marine, or on land, but many members of rhodophycidae are marine. Then the thallus, or the plant body of bangiophycidae, is simple, unicellular, branched, branched but never shows any pseudoparenchymal structure, while that of rhodophycidae is complex, shows aggregation of filaments, and it has a pseudoparenchymal structure. Just to take you back a little bit, you can see this gelitium, which is a member of rhodophycidae. It has that structure that enables it to look like a plant. So this structure is what we call the pseudoparenchymal structure, because it is from it that these other things grow from. Then bangiophycidae members are uninucleate. Many of the members of rhodophycidae are multinuclear, and they have as many nuclei as possible. Then their growth is diffuse. We said what makes it diffuse? The zygote undergoes meiosis. The whole zygote breaks down to form carpospores, while the ones of rhodophycidae, their growth proceeds with the help of a single apical cell. We said the carposporangium, the apical cell of the gonymoblast forms the carposporangium that forms carpospores. Then each cell has a single stellate chloroplast with a single pyrenoid. Then here each one has many peripheral discoid chloroplasts with no pyrenoids. Then the zygote undergoes meiosis to form carpospores. Here we see the zygote undergoes mitosis to form a gonymoblast. The carpospores on liberation form the chantarisia. Here we see they form the tetrasporophyte. Then their life cycle has two haploid phases and one diploid. Here we say two diploid phases and one haploid phase. Then each zygote forms 4 carpospores. Each zygote forms 1 carpospore. Then the gametophytes develop from the chantarisia. Here the gametophytes develop from the tetraspores. Then their gametophytes are bisexual. Their gametophytes are unisexual. Okay, so we look at another algal division, which is chlorophyta. This division is also known as isocanthae, and it is a very diverse group of algae, and it is found in freshwater or land, most especially. So what are some of the characteristics? Number one, they have flagella, and the flagella are of the same length. That is why it's called isocanthae. Remember, we said flagella of the same length is called isochord, and the one of different length is called heterochord. So these flagella are of the same length, and they are of acronomatic type. So number two, they have both sexual and asexual reproduction. Their photosynthetic pigments include chlorophyll A, chlorophyll B. They have some carotenes and xanthophylls. Then their food storage reserve is true starch. Then their cell walls have cellulose. And their reproduction is oogamous, and they resemble plants, but many of them are found in fresh water. Examples include Clinodomonas. We have Corella here, Spirogyra, Odegonium, and Volvox. These are some of the examples of chlorophyta members. We have this vulva, we have cladophora, we have this hydrocytone. This is a colony of hydrocytone, and this is bistromatic. Okay, so another group of algae is charophyta, also known as the stoneworts, and it has only one order, which is charels, and this charels is divided into 8 genera, and the most common genera are Chara and Nitella. What are some of the characteristics? Number one, they are macroscopic. They are easily seen with our naked eyes. They are macroscopic and they look like plants attached to the bottom by rhizoids. Then their plant body consists of an erect axis, and this erect axis has nodes and internodes. And with whorls of branches, groups of branches. But these branches have limited growth at the nodes. What does that mean? This is an example of a stone wood. You can see here we have very many groups of branches coming from nodes, and in between the nodes we have the internodes. So that is what the characteristic means. Number 3, their sexual reproduction is oogamous. And unlike other algae, their multicellular reproductive organs are surrounded by a layer of sterile tissue. Remember, once we take you back previously, we say that um, many members many members of the algal division have uh, they are unicellular, and if multicellular they lack a zone of sterile tissue. But this group is unique in such that it has a layer of sterile tissue surrounding its reproductive organs. Then the anthrozoids, or the male motile gamete, is elongated and it is made up of two flagella, so it has a biflagellate structure. And the other thing is their zygote develops into a protonema, and that protonema is simply an intermediate stage. So it is from the protonema that the thallus will develop, and their thallus is also encrusted with a lot of calcium carbonate, which makes them extremely hard. Hence the name stone warts. Then this is just an illustration of the stone warts. The next group is bacillariophyta. So bacillariophyta is made up of unicellular or colonial. That means you can find one cell, or you find many cells clustered together, what they call colonial, and they occur in both freshwater and marine habitats. And they are very prominent in the soil and at the bottom flora of lakes. What are some of their characteristics? Number 1, their cell wall contains silica. And their cell wall is made up of two overlapping halves or shells known as frustules. Then the frustules are divided into two, the upper part and the lower one. The upper part is what we call the epitheca, and the lower part is what we call the hypotheca. Then their cell walls are resistant to decay, and they form deposits of diatomaceous earth, or what we call kiesilga, which is found in lakes and ocean beds. We have already discussed the importance of diatoms or diatomaceous earth or kiesilga in our video talking about importance of algae. If you've not watched it, please do so. And because they have an extensive skeleton made up of silica, many of them have an extensive fossil record compared to any other non-algal group. Actually, diatoms are very important in what we call paleolimnology, which we shall discuss later. Then their major photosynthetic pigments are chlorophyll A, chlorophyll C, alongside other xanthophytes. And their food storage products are fats and chrysolaminarin. Then their motile states possess a single pantonematic flagellum, and their sexual reproduction is by formation of axospores. Then the diatoms are divided into two orders: centriales, or what they call the central diatoms, and the penales, or what they call pinnate diatoms. And there are some differences. For example, the centriales have radial symmetry, the other ones have bilateral symmetry. The centriales are non-motile, while the penales have that gliding motion. The centriales lack the raphe, while the penales have an longitudinal fissure or slit called raphe. Then the centriales have only woogamy as a mode of sexual reproduction. The other ones also have isogamy. Many of the centriales are found in marine water, while the penales are found in freshly water. So here I just have an example of two centriales. This is Cosmidiscus and Rhizoselenium. So this is how they look like. Okay, another group is Euglenophyta, or what they call the euglenoids. So what are some of the characteristics? Number one, they are unicellular, uniflagellated organisms. They are made up of one cell and they are made up of one flagella. Number two, their major photosynthetic pigments are chlorophyll A, chlorophyll B, carotenoids like beta carotene, lutein, neoxanthin, astaxanthin, and anthraxanthin. And these pigments are also found in chlorophyta. Then the ones that have pigments are autotrophs or facultative heterotrophs. They can feed on other organisms or they can feed on already manufactured food, while the unpigmented forms of euglenoids are obligate heterotrophs. Then euglenoid cells are plastic. What does that mean? They lack a cell wall. And that lack of a cell wall enables the heterotrophs to be able to feed by phagocytosis. Then since they don't have a cell wall, their outer body is protected by what we call a pellicle. A pellicle. They have one, two, or three pathognomonic flagella result arising from the invagination of the interior of the cell. Then their major food sources is paramylon, which is made up of polysaccharides. Some of the common examples are colassium, euglena, Tripitalia, and Fucus. However, I need to emphasize that the taxonomy of euglenoids is contentious. It is ambiguous because of their motility and heterotrophic nature, because many of the algae are autotrophic and non-motile, but euglenoids are able to move and many of them are able to feed on already manufactured food. Okay, so we need to look at another group of algae, and those are the dinophytes or dinoflagellates or pyrophyta. So what are some of their characteristics? Their photosynthetic pigments are chlorophyll A, C, have some carotenoids like dinoxanthin and pyridine, and they appear brown because of pyridine. Then um, they are photosynthetic pigments. They are photosynthetic organisms surrounded by one layer. Uh, they are photosynthetic organisms surrounded by one. They are photosynthetic organisms having chloroplasts surrounded by one membrane. You know that chloroplasts are down membrane organelles. Um, then the outer membrane is their outer membrane or the outer cell membrane is a continuation of the nuclear envelope. You can see how unique they are. Then their major storage product is starch. They store starch. Then the cells are biflagellate. They have two flagella with one band shaped and the other is achromatic. Then their nucleus is permanently condensed, permanently condensed chromosomes. So their chromosomes are in interphase, and this stage is called being dinokaryotic or mesokaryotic. And since it is a nucleus that has the chromosomes, so they have a dinokaryotic or mesokaryotic nucleus. Then most membrane-bound organelles contain structures called pustules. And these pustules look like contractile virtue, but they don't show any function of contractile activity. Then the mitochondria has tubular cristae. Then most members have ejectile trichocasts. However, the function of the trichocast is not yet known. Then dinoflagellates have what we call cellulosic plates that forms an armor around the cell, and this is one of the basis of their taxonomy, and many of them can emit light, so we can say that they are bioluminescent. They are bioluminescent. They are bioluminescent. So we need to look at the structure of a dinoflagellate. So a dinoflagellate is made up of a transverse furrow, and that transverse furrow is what we call a groove. It's called a transverse furrow or groove called a cingulum. So it has this transverse furrow, it cuts across It's called a transverse furrow, called the cingulum or the girdle, and it divides the outer wall into the forward portion and the downer portion. And the forward portion is what you call the epicone, and the downer portion is what you call the hypocone or the hypotheca. Parallel to the cingulum is this structure called the sulcus. And the sulcus is where the two flagella arise. I mean, the junction between the cingulum and the sulcus is where the two flagella arise. And this is how a dinoflagellate looks like in real life. This is how it looks like. Okay. Then the Then you need to look at another group, which is phycophyta or brown algae. So the phycophyta are what we usually see as large sea weeds known as racks and kelps. They are very long and large, and actually some of them can even grow up to a length of 20 meters, and they exhibit a high morphological diversity compared to any other algae. So when you hear of things like racks and kelps, just when they're talking about brown algae. So what are some of their characteristics? Number one, their photosynthetic pigments include chlorophyll SC, carotenes, and xanthophytes. Major food trade products are laminarin, mannitol, and focusan. Their cell wall is made up of alginic and fucinic acid. Then their motile stages appear shaped with two flagella, one acronemmatic and one pantenomatic flagella arising from each side. Then their mode of reproduction and life cycles shows a comparable high degree of specialization, and their sexual reproduction usually mass. Then the organization of their thallus is more advanced compared to any other algae, and their growth is parenchymatous in many species. That is why many of them look like plants. Some of the examples are, we have Ascophyllum, Itocarpus, Fucus, Laminaria, Microcystis, Nereosocytis. We have Pelagophycus, Pelevetia, Pocelsia, and Sargassum. So here we have an example of Fucus and we have an example of Itocarpus, but you agree with me that these look like plants, yet they are not plants. Okay, so we look at the next division, which is Cyanophyta. Cyanophyta is that group that is also known as blue-green algae. They were traditionally not algae. Yes, actually, they were traditionally classified as algae, but because of their prokaryotic nature and new new taxonomic tools like DNA sequencing and genome sequencing, they have now been moved from algae to bacteria. However, they still possess some characteristics that make them look like bacteria. Number one, they have chlorophyll, and their chlorophyll is found within the lamellae instead of the chloroplasts. And when they are to move, or when movement is to occur, they move by gliding. The other thing I forgot to tell you, Cyanophyta was previously known as Myxophyceae, and the name has just changed recently. They form a mucilaginous sheath, and because they form mucilaginous sheath, they are previously known as Myxophyceae. So they don't have any flagella. Their major storage products are cyanophycin and cyanophycin starch. Then their photosynthetic pigments are phycocyanin and phycoerythrin, which gives them the blue-green color appearance, and they have some carotenoids such as myxoxanthin and myxoxanthophyll. I've already said they lack flagella. Then they possess enlarged specialized cells called heterocysts, and we say the heterocysts contain the enzyme nitrogenase, which nitrogenase is able to convert atmospheric nitrogen into compounds that can be absorbed by cells for protein synthesis. Then we talked of homogonia. We said homogonia are short motile gliding filaments for reproduction. So the homogonia are unique to xanthophyta. Then they don't have any form of sexual reproduction, and they reproduce asexually by forming endospores, ectospores, akinets, and hypnospores and homogonia. So we can see some of these. You can see here we have our heterocyst containing nitrogenase. This anabaena had formed akinets. This is anabaena. Anabaena is an example of cyanobacteria. You can see akinets here. You can see heterocysts and you can see vegetative cells. And remember, we said akinets are formed when vegetative cells are converted into spores. This is an illustration to show us homogonia. This is an example of oscillatoria, showing oscillatoria filaments. Oscillatoria filaments. So we need to look at some of the why blue-green bacteria has been transferred to algae. Number one, their cell membrane. Their cell membrane. No, but absence of membrane-bound organelles. So because just like bacteria doesn't have membrane-bound. Doesn't have membrane-bound organelles. So because of that, scientists decided to say, let us keep this in the bacterial division. So the first one is absence of membrane-bound organelles. Number two, their cell wall composition. So their cell wall composition is similar to that of bacteria in such a way that they both have a peptidoglycan layer, or what we call the mucopolymeric. Number three, their cell division, that they both reproduce by cell division, which is usually by binary fission. Mitosis and meiosis never occurs. Ribosomes, they both have 70S ribosomes. Then they both have a slimy sheath. Both bacteria and cyanobacteria have a slimy sheath called the glycocalyx. Then they both form in foldings known as mesosomes. And science has shown that in cyanobacteria, the mesosomes appear to be the photosynthetic apparatus where photosynthesis takes place. Then they both form endospores, and endospores are used for asexual reproduction, and they both have the ability to convert atmospheric nitrogen into other forms that can be used for protein synthesis. So with those, scientists decided to transfer them to bacterial kingdom. So what are some of the differences between cyanobacteria and bacteria? Number one, cyanobacteria has phycocyanin and phycoerythroid, while bacteria does not have them. Number two is the absence of bacteriochlorophyll. That bacteria has bacteriochlorophyll, while cyanobacteria doesn't have the bacteriochlorophyll. Then cyanobacteria doesn't have any flagellated form, while some bacteria have flagella. Cyanobacteria doesn't have this extra chromosomal DNA called plasmids, while bacteria has the plasmids. Cyanobacteria forms heterocysts, bacteria doesn't form heterocysts. Cyanobacteria stores cyanophycin, bacteria doesn't store cyanophycin. Many bacteria have pili, cyanobacteria doesn't have pili. Cyanobacteria doesn't carry out any fermentation, many bacteria can carry out fermentation. Cyanobacteria doesn't have any form of sexual reproduction. Bacteria has different forms of sexual reproduction, conjugation, transformation, and others. Then cyanobacteria can form homogonia. Bacteria cannot form homogonia. So we need to compare cyanobacteria with other algae. Except cyanobacteria, all other algae are eukaryotic. That means they have a nucleolus. Cyanobacteria doesn't have the nucleolus. Cyanobacteria has circular DNA. This one have linear DNA, which is associated with proteins called histone. Cyanobacteria cells are smaller, the others are relatively larger. These ones don't have any down membrane bound organelles, these ones have down membrane bound organelles. Their cell wall is made up of a peptidoglycan. This one is made up of either cellulose, chitin, silica, and others. Cyanobacteria don't have any flagella. Many species of algae have flagella. They have bioloproteins, phycocyanin and phycoerythrin. Except rhodophyta, the rest don't have bioloproteins. Many cyanobacteria have the slimy sheath. No other algae has been noted to have the mucilaginous sheath. These ones have mesosomes. Mesosomes are absent in eukaryotic algae. These ones can form, cyanobacteria can form endospores. Eukaryotic algae cannot form endospores. They don't have any sexual reproduction. These ones can reproduce either by isogamy, anisogamy, or ugamy. These ones form gas vesicles that enable them to float in water. No other algae has been noted to form gas vesicles. Cyanobacteria can convert atmospheric nitrogen using near nitrogenase. No other algae can carry out nitrogen fixation. Okay, I need to remind you, if you need these notes or you need trial items, please click the link in the description box and it will take you directly where to access. Then please don't forget to like, subscribe, and follow us on our social media handles. So we look at the last group, which is xanthophyta. So members of xanthophyta are also known as yellow-green algae, and they are found in fresh water. Others are terrestrial, while a few are marine. So what are some of their characteristics? Number one, they have motile cells with direct tinsel flagellum that is anteriorly located and whiplash flagellum that is posteriorly located. So the chloroplasts contain chlorophyll A and C, which makes them appear yellowish green, but they also contain some carotenoids like diadenosxanthin, heteroxanthin, and vitoroxanthin. Then their chloroplasts, their chloroplasts, their chloroplasts contain thylakoids in groups of 3, and in many genera there is a single pyrenoid in the chloroplast. Then they also have an eye spot similar to what we had seen earlier. Then their chloroplast is enclosed by 2 membranes which move from the endoplasmic reticulum, while the outer membrane is continuous with the outer nuclear membrane. Then their food storage reserves are mannitol, then some glucans, and others have lipids. Then their vegetative and asexual reproduction takes place by either fragmentation, that is vegetative, and asexual reproduction by production of zoospores, planospores, or forming akinets. And their zoospores don't have a cell wall, they are biflagellate with an anterior large flagellum and a posterior small whiplash flagellum. Then sexual reproduction is only known in members of Vaucheria, Tribonema, Botridium, and in Vaucheria it is oogamous, while others they can have both isogamy or oogamy. So we need to look at some of the examples of xanthophyta. We have here Botridium, and this is Vaucheria. And that is what we had for you today. Till next time.